So I'm assuming that most people listening will have more knowledge of ketogenic diets and ketogenic metabolism than knowledge of sleep. And so I'm going to spend a little more time going over some basic sleep physiology, and then we'll integrate these concepts. Two focal points from which we could view sleep effects are the metabolic and the neurological or cognitive. And interestingly, the same is true of ketogenic diets. So most of what I talk about will be centered on these. The mystery of what sleep is actually from a physiological perspective isn't really known for sure. That's not to say that there aren't people with theories, good theories with a lot of support. There are those, and I'll mention some of them, but it's not a question I'm going to even begin to try to answer here. What we do know with certainty is that sleep is needed, and we know that because we know some of the consequences of not getting it, and conversely, we know some of the benefits it provides. We also know that sleep is regulated homeostatically, meaning just that there's a drive for sleep, and that ensures that we get enough and we don't get too much. So one way that we can tell that sleep is homeostatically regulated is that the longer we're awake, the more irresistible sleep becomes. This pressure builds up, and it builds up essentially linearly the longer we're awake, and then it falls off when you go to sleep. But it falls off faster than linearly, and we'll talk about why. But to understand that, we need to talk about some of the measurable characteristics of sleep. So if we want to talk about the effect of a particular intervention on sleep, we have to have a way to evaluate it, not just the subjective way we normally talk about sleep. And there are different ways to do that. Duration is the most straightforward one. It means how much time elapsed from when you went to sleep until you wake up. But a nuance there is that from the moment you initially go to sleep until you wake up for the day, you might not actually be asleep the entire time. So that's where we talk about sleep efficiency. And that can be measured either as a proportion of time asleep or the number of wakings, for example, are typical ways. Sleepiness in the daytime is also a qualitative measure or a quantitative measure, it can be, of a qualitative part of sleep. So there are two typical ways that we measure that. One of them is simply by asking people to imagine themselves throughout the day in situations like stopped at a red light or curled up on a couch with a book. And you're asked to rate how likely it is that you would fall asleep in that situation. So that's somewhat subjective. And so a second, more accurate way is the Epworth scale, in which you actually ask subjects to come into the lab and try to fall asleep. And then you measure how long it takes them to if they do achieve it. So the faster that you fall asleep, the more sleepy you're considered to be. Sleep stages are important, and I'll go into them in more detail. The basic idea is that we are measuring brain waves during sleep, and we find that we fall into these periods of characteristic patterns. And so when we're evaluating an intervention, we want to know how it affects those patterns. That's called sleep architecture. And then with circadian rhythms, what we're talking about is looking at a variety of physiological processes and how they vary throughout the course of the day in a cyclic pattern. So we can measure changes in these cycles that different interventions might induce. Okay, so let's talk about sleep stages. It's pretty easy to tell when an animal is asleep or awake because of behavioral things like being very still or in a specific posture. For example, humans would normally be lying down with their eyes closed. Also, lack of responsivity is important. So if you're making some stimulus like noise and the person doesn't respond, that is a cue that they may be, or a clue that they may be asleep. But we can also see differences if you look at brain waves using electrodes, an EEG from the scalp. Once you start looking at the brain waves, you'll start to see that there are further distinctions that can be made during sleep itself. The primary distinction that we make is between REM, which stands for rapid eye movement because our eyes are moving rhythmically, and non-REM. And we cycle between those, usually starting with non-REM at the beginning of the night. So REM is physiologically in many ways like being awake. The brain is really active. But the primary difference is that in REM sleep, your motor neurons are disengaged, and that's presumably so that we don't act out our dreams. Vivid dreams are a characteristic of REM sleep. There can be dreaming in non-REM, but it's rarer. And if you don't have that muscle disengagement, it can be very dangerous. There's a sleep condition called REM behavior disorder in which motor neurons are not properly turned off. It's thought to be neurodegenerative, and it's very dangerous. Now, non-REM itself can be split into light sleep and deep sleep. And they have further subdivisions that we won't go into very much. But deep sleep, it's mostly, I think, called that to reflect the fact that the deeper the sleep that you're in, the less responsive you are, the more intense the stimulus has to be to wake you up. But it also corresponds to whether or not the EEG has a lot of delta waves in it. Delta waves are relatively slow, so we call deep sleep slow wave sleep. And what's happening in slow wave sleep is that your neurons are rhythmically turning off in a coordinated way. It's called coordinated neuronal silencing. And this produces a high amplitude wave. So here I'm showing a kind of typical order, but it's not strictly ordered like that. In this diagram, it's called a hypnogram. There's a typical pattern of sleep stage transitions for a healthy adult over the course of the night. You can see we go through cycles of hitting various stages. The most important things to note are that most of the deep sleep, in the blue, happens in cycles at the beginning of the night. There can be cycles with deep sleep later in the night, different from here, but most of the slow wave activity occurs in the beginning. And then most of the REM happens in the last half of the night. The differences between stages 3 and 4 here have to do with the intensity of the delta wave activity. 3 and 4 are arbitrary cutoffs, and we can also look at it less discretely. So here we have another hypnogram at the bottom in blue, and I've circled the areas with stages 3 and 4. The hypnogram is derived in part from the red graph above, which is showing the spectral power of the delta waves. So you can see that these divisions into stages 3 and 4 come from amplitude cutoffs, but you can also see, if you look at the area under the curve, that most of the intensity happens in the first couple of cycles. And so now when we go back to the concept of sleep homeostasis, you can see that the nonlinear way that sleep drive falls off corresponds with slow wave activity. And this is one of the reasons that we associate sleep homeostasis with slow wave activity in particular. Slow wave activity and slow wave sleep are important for various things. And one of the things that they appear to be important for is metabolic or endocrine level things. So we have a correlation, for example, between body mass index and slow wave sleep. But we also experimentally can show that a deficit in slow wave sleep can result in glucose intolerance and insulin resistance. Most of what we know about slow wave deep sleep is connected to cognition. So slow wave sleep plays a role in memory consolidation, for example. We also have a number of associations in certain conditions where cognitive deficits appear. So for example, in Parkinson's and in Alzheimer's and even just in aging. Some researchers have looked at the amount of slow wave sleep and made an association between the degree of cognitive deficit and the degree of lack of slow wave sleep. And then experimentally, some researchers have tried to see if efforts to try to increase slow wave sleep could have a benefit on cognition. And they found that insofar as their efforts to increase slow wave sleep were successful, there was some association with improvements to cognitive deficits. There's also a phenomenon called use-dependent recovery, where the areas of the brain that are most active while awake are then the ones that have the most slow wave activity in the next sleep opportunity. So that could be sensory motor. For example, if you stimulate one part of the body or move it around a lot, then the associated sensory and motor neurons in the brain will have more slow wave sleep than the non-stimulated parts. And then likewise, if you force one part of the body to be still, that would mimic, for example, your arm being in a sling, then there would be less slow wave activity in the associated areas of the brain than otherwise. And so that's thought to show that muscle atrophy from non-use, while part of it has to do with actual muscle tissue shrinking, part of it is actually a neurological non-use kind of decay. But use-dependent recovery can also be cognitive, where it's linked directly to learning. And more generally, the more energy that your brain or a part of your brain uses, the more subsequent slow-wave sleep there is. This diagram's from a paper exploring the idea that the enhancement of slow-wave sleep might be used therapeutically to improve cognition. So they identified several ways that we can increase slow-wave sleep that would be candidates for this kind of experiment, and one of them is by increasing energy use in the brain. So we now have the foundation of our map. Adequate slow-wave sleep can preserve cognitive function and glucose tolerance, and one of the things that contributes to slow-wave sleep is energy use in the brain. A whole other aspect of sleep is the circadian aspect. Circadian effects are biological patterns that change with the cycle of light and dark. So this graph, for example, shows how cortisol release is circadian. It peaks just after waking, it drops throughout the day, and reaches its lowest point toward the beginning of the night. The darker black line here showing a normal circadian rhythm of cortisol, and we can see that if you completely sleep deprive the subjects, which is shown in the white squares here, yes, it reduces the full amplitude of the drop in cortisol, and the height in cortisol after waking is persisted slightly longer, but it doesn't abolish the whole circadian nature of the cycle. On the other hand, some things look circadian, but in fact are a product of the sleep-wake cycle. So, for example, growth hormone release is like this. If you looked at the beginning of this graph, where there's this big peak in growth hormone at the beginning of the night, and it's mostly low for the rest of the time, you might think that growth hormone release is circadian. But if you don't actually sleep at night, it turns out that that growth hormone release doesn't peak like that. It doesn't peak again until you are allowed to sleep. And it doesn't matter when that sleep happens, even if it's in the day, the peak will happen then. So, in fact, it's not a circadian rhythm, it's a sleep-wake, it's sleep-dependent. So if you're really paying attention, you'll notice that this peak here in the first half of the night corresponds to when slow-wave sleep is the highest. And that's thought to be actually a causal dependency. Pulses of growth hormone come right after pulses of slow-wave sleep. And so we think that growth hormone is directly dependent on slow-wave sleep. Now, many processes are periodic in this daily pattern. Many tissues and organs and hormone-releasing organs have their own rhythm with which to coordinate their action through circadian signals. So they take signals from the brain telling them where they are in the light-dark cycle, but they will actually make their own cycles based on that. And so that means that if they get misaligned from strangely timed sleeping, for example, strangely timed activity or eating patterns, systems that used to be in sync with each other will get out of sync. So to take just a very simple example, if you create a circadian misalignment in subjects and give them breakfast at the same time with the same kind of food that you gave them when they were in alignment, the glucose and the insulin response will be much larger with a circadian misalignment than otherwise. So we'll add this to the graph. Circadian alignment can contribute to healthy glucose tolerance. When you go from a high carbohydrate diet to fasting. At the beginning, you'll use up glucose from your glycogen stores. We can call that phase adaptation. It takes just a couple of days. And once glycogen is depleted, you can start relying almost exclusively on fat stores. Of course, there will always be some lean mass loss with fasting. But when there's a lot of fat to metabolize, several mechanisms can come into play to preserve lean mass. This fat phase can be considered a steady state in humans because even lean people have weeks worth of fat calories. So when that gets used up, then you will go into this late stage starvation where you have to start tapping into lean mass in a big way, and that very quickly leads to death. I want to focus on the steady state in the middle after adaptation and before real starvation kicks in as our best comparator for a ketogenic diet. This is a diagram from Cahill, a pioneer in the study of fasting metabolism. When he says normal here, he means high carbohydrate, and when he says starvation, he means that steady state. And it's just showing that the brain uses ketone bodies now as compared to using almost exclusively glucose in the high carbohydrate condition. The functional reason for ketone bodies is that most fat doesn't get through the blood-brain barrier. And that's thought to be because fat oxidation generates a lot of oxidative stress, oxidative products. And a lot of the brain tissue is made out of polyunsaturated fats, which would be very vulnerable, susceptible to oxidative damage. So rather than using fat directly, using ketone bodies, a product that part of that oxidation has already occurred, is a much more safe way to run the brain. There's also a nice feedback loop when you're fat adapted, such that the more fat you're using for energy, the more it spares protein. Protein is needed when you're not eating carbohydrates as one of the substrates that glucose is made out of by gluconeogenesis in the liver. So in one way that this spares glucose is simply that many tissues can use either fat or glucose, and so if the glucose isn't there, they will use fat. But there are also other ways in which muscle tissue can be spared. So for example, glycerol is one thing that triglycerides are made of. Triglycerides is the form of fat that we mostly eat or get from our stored fat tissue. It's got 3 strands of fat tied together by a glycerol backbone. And so when you take off those fat strands to burn them, you're left with glycerol, which very conveniently can be used for gluconeogenesis. So the more fat that you're burning, the more of that you have available. Similarly, one of the ketone bodies, acetone, is actually a substrate for gluconeogenesis. So the more ketogenic you are, the more of that you have available. Ketone bodies themselves, from a signaling point of view, seem to give the signal for the body not to tear down protein. So that's another anti-catabolic mechanism. And finally, I wanted to just mention the Cori cycle, which was pointed out also by Cahill, where what's happening is you've got glucose that you made in the liver and you send it out to the periphery and it gets oxidized. And one of the oxidation products is lactate. Lactate gets sent back to the liver and then can be it's a substrate for gluconeogenesis, so it can be made back into glucose. Now, of course, it's not a perpetual motion machine. You need to add something, but it turns out that what you need to add to make that happen is simply energy that can be provided in the form of fatty acid oxidation. So that's one way that you can use fat to provide glucose via recycling. All right, so here's our first study on fasting, the effect of fasting on sleep. It's a small study, but it's informative. They took 10 healthy men and they fasted them for 4 days. Here's what happened to slow wave sleep. You can see the effect took a couple of days to fully appear, but slow wave sleep was increased. Similarly, we see that REM sleep went down slightly over the course of a few days. Here's a second human study. It actually wasn't full fasting. There was a restriction to 10% calories. That's enough to start that fasting metabolism. The caloric restriction phase is in the black bars. If you look at panel A, you can see that they only went 2 days, unfortunately. So we don't expect necessarily to see the full effect that we might get if they went longer. Panel B shows that REM sleep went down, but it went down by such a small amount that it's not even statistically significant. Now, non-REM did go up significantly. Panel C shows that it's all from stage 4. This panel E at the bottom shows an increase in growth hormone, which you might expect because, as we've seen, growth hormone pulses are very tightly connected to slow-wave sleep. Now, growth hormone increases in it are a robust finding in fasting, and we do talk about that a lot in the ketogenic community, but I rarely see that explicitly connected to sleep, so it was nice to see that here.